My name is Colleen Dansereau. I'm the Vice President of Emerging Discoveries in the Gene Therapy Operation Program at the Boston Children's Hospital. And I'm going to speak to you today about the nursing perspectives in gene therapy. I'm going to start by doing a little bit of a primer on what is gene therapy. So gene therapy is the introduction of a new or a modified copy of a gene. It could be the turning on or off of genes to avoid or ameliorate symptoms of a disease. It could be the replacement of a disease-causing sequence of a gene with a healthy copy of that sequence. And unlike traditional medication, it actually works on the underlying genetic problem or the root cause of the defect and not just the symptoms. There are several types of gene therapy, and so one of the very basic parts is that there's germline, and germline gene therapy would be adding or subtracting genetic material which could into the egg or the sperm cell, which could be inherited or passed on to the offspring. And subsequent to that, any modification you made to the egg or the sperm cell would actually be present in any prodigy that came from that egg or sperm cell. Somatic gene therapy, which is what we're going to talk about today, somatic gene therapy would be any changes made to the gene or the genome would only affect the person who we are modifying their cells or their body. So those cannot be passed on to the offspring. So in genetic diseases, if we were to do gene therapy on the patient, if that patient went on to have offspring, the offspring could very well still carry the genetic either predisposition or the genetic code for that disease. So that's an important distinction. And then there are two subtypes of gene therapy. One is in vivo, and in vivo gene therapy, the gene is actually transferred directly to the patient and the patient's cells while those cells are still in the body. And then ex vivo, which is what we're going to talk about today. Ex vivo is the genetic manipulation is done outside of the body. So we would take the patient's cells, be that hematopoietic stem cells, CD34 cells, or T cells for CAR T. Those are taken out of the body and then manufactured or genetically manipulated in a laboratory and then transferred back to the patient. So within the ex vivo gene therapy space or the modalities, there are several now types of gene modalities. The first being gene transfer or gene addition. Gene silencing is another one, and then a more precise and more refined types of ex vivo gene therapy have become gene editing and base editing. In order of the listing, there is the degree of specificity with the actual gene modification. Gene transfer or gene addition was one of the first types of ex vivo gene therapy, and that is gene addition is used to treat conditions caused by a variation in a single gene or monogenetic. So this is a one-time therapy and involves the insertion of a functional copy of a missing or faulty gene into the cell nucleus using a viral vector or a non-viral genetically engineered vector as the delivery vehicle. The goal of this therapy is for the new gene to produce a protein that was previously missing or inadequately expressed. Gene silencing is transferred via viral vector to cause a post-transcriptional gene silence. It inactivates specific messenger RNA to prevent the expression of a gene. The mRNA of a particular gene, which ordinarily would be expressed or turned on, is actually suppressed, knocked down, or turned off. When the mRNA is knocked down, it can no longer create an active protein at the same level as it was before. Gene silencing has become a common method used in certain monogenetic conditions, and the one most commonly used lately is sickle cell disease. Gene editing, as I said, is a more precise type of genetic modification. Gene editing is a precise technology used to add or remove or alter specific DNA sections. It uses a CRISPR technology to find where the faulty sequence is and a Cas9 proteinase to make cuts to the DNA at the specific spot where that is malfunctioning. After cutting the DNA, the cell will naturally repair itself. By disrupting unwanted sequencing elements or by copying pasting in the new sequences to fix the faulty sequence, gene editing can lead to a permanent genetic change. Cells will normally function normally and reverse the disease process. Base editing, again, more refined, base editing targets a single point mutation and creates a precise change in the genetic code. These changes can silence a disease-causing gene or help activate a specific gene. Base editors don't cut the double-strand DNA as in editing, but instead deliver an additional enzyme to a desired sequence to change the base on one strand. Finally, cellular repair proteins recognize the base change and convert it to a permanent mutation. Then the cells can take on a new function normally, and conditions caused by a specific mutation potentially could be cured. In order to get the gene into the cells and gene addition or gene transfer, we need to use a vector. And a vector is a vehicle that transports the gene into the cell nucleus. We use viruses because, as people can appreciate, viruses have a very have great virulence and have great ability to permeate the cell membrane and to actually get into the cell nucleus. Viruses that we use could be HIV, an adeno-associated virus, lentiviral vectors, or gamma retroviral vectors. We take advantage of the ability to permeate into the cell nucleus, but what is done is the viruses are modified to remove the infectivity or the replicating infectious particles. So we harness kind of the engine, if you will, but modify it in a way that it can't replicate it and it's not infectious. The gene-based editing technologies use a CRISPR-Cas9 technology, and the CRISPR is a guide RNA strand that finds specific DNA strands that need to be edited. The Cas9 is a protein that is a nuclease enzyme that has the ability to make the DNA break once it's been located by the CRISPR at the location. All of these ex vivo technologies have the same starting material, and the starting material is the cell source, being hematopoietic stem cells or CD34 cells. In the case of a CAR T cell, the differentiation would be we would be collecting lymphocytes. For ex vivo gene therapy, collecting HSCs, we would use two agents to mobilize the HSCs out into the periphery. The first drug is GCSF, or granulocyte colony stimulating factor, or palixifor. Both of these agents are given, and they actually mobilize the stem cells from the bone marrow niche out into the periphery so that they then can be collected via leukapheresis. Leukapheresis is the preferred mechanism to collect stem cells for gene therapy. If there is some contraindication for leukapheresis, and one of the reasons you could perhaps not do leukapheresis on a patient would be the size. So in small infants, less than 5 kilograms, it may not be feasible to do leukapheresis. In that case, we can do a bone marrow aspirate, and in the bone marrow, we can certainly harvest stem cells through a bone marrow aspirate. And in addition to collecting stem cells for genetic manipulation, we'll also do a collection of backup or a separate collection of stem cells that will be stored in off to the side or in the freezer, and they are unmanipulated. And I'll go into why we would use those in the future. The genetic manipulation, if you will, or the transduction of all of the processes that I outlined as ex vivo gene therapy treatment modalities all start the same way. So the collected apheresis product and/or bone marrow, if that be the case, are purified in the laboratory, and so all of the other perhaps cellular debris or cellular material is removed, and you have isolated CD34 cells. Those cells are put into a cell culture medium with the vector or the gene editing technology, depending on the type of modality you're doing. The medium is actually a fluid that has different types of growth hormones, glucose, and other materials that will keep the cells healthy and happy, actually. The culture medium is changed at 24 hours. And then more vector, it's rinsed out and replaced with more vector or gene editing technology, more CRISPR-Cas9 zinc fingers is added. At 72 hours, the stem cells are removed from culture, and then they are tested for the presence of the vector or the gene edit that was done, as well as sterility, cell count, cell number. This is a schematic of what that process looks like. So as you can see in number 1, the cells are collected from the blood or the bone marrow. The patient's target cells are isolated as the CD34 cells. The gene therapy is packaged into a delivery vector, such as an engineered virus. This would be for gene transfer, gene addition. You could supplement if you were using gene editing. You would use the CRISPR-Cas9 and the CRISPR and the Cas9. Then the vector is introduced into the cells. The cells take up that vector. As you can see in number 5, it's present in the center. And then those genetically modified cells are returned to the patient who may require specific conditioning. And we'll get to that in a second. The patient preparation is that, as I said, we do collect a backup of stem cells that are stored unmanipulated, or no gene manipulation. That is in the event that anything goes wrong during the process, whether the patient doesn't engraft, the genetically modified cells don't happen to take in the patient. We have ability to rescue that patient, if you will, with a backup cell. The patient undergoes a standard of care pre-transplant bone marrow transplant assessment per your institutional standard, admitted to the bone marrow transplant service, and then the patient receives a myeloablative conditioning, which makes space in the bone marrow to clear out whatever existing stem cells are left behind and make room for the new genetically manipulated cells. The myeloablative conditioning of choice is generally busulfan as the agent of choice, and some, depending on the condition, may also add fludarabine. For CAR T cells, they use a lymphodepleting chemotherapy, and that is actually Cytoxan and fludarabine. The gene therapy product infusion is administered intravenously through a central line. It follows the same policy and procedure institutionally as your blood transfusion, with a pre-medication monitoring and documentation. Its administration technique is protocol and/or product specific. So as many of these ex vivo gene therapy products are now EMA or FDA approved, they are actually off protocol, but your protocol and/or your product label will dictate how it can be given. But generally, it's given either an IV drip, an IV push, or via a volumetric infusion pump. The one constant is no filter is used in any of these products. And it also is given, its intent is a curative, so it's given as a one-time single infusion. So the product is infused via, given as a one-time single infusion. The other piece of note is that it's an autologous product, remembering that the CD34 cells came from the patient, so unlikely to trigger an infusion reaction because it's an autologous product. I'm going to use an example, as we did, of ex vivo gene therapy. So this, I'm going to outline the steps and kind of the scientific rationale and how gene therapy works in sickle cell disease. So this is gene therapy for sickle cell disease. This happens to be a patient we treated at Boston Children's, one of our first patients we treated with gene therapy for sickle cell disease. But remembering kind of the steps as to why you would use in ex vivo gene therapy. Sickle cell is a great indication. It's a single gene deletion, so it's monogenetic. BCL11A is the gene responsible for making hemoglobin S, or adult sickle hemoglobin. In utero, we make hemoglobin F, and that after birth, at about 6 months of age, you internally make a switch, and fetal hemoglobin turns off, and hemoglobin S starts. Hemoglobin F doesn't sickle. It's well documented that sickle cell patients who have a higher hemoglobin F have less severe sickle cell symptoms. So with the hypothesis that if you shut off BCL11A, which causes hemoglobin S, then your body would continue to make F, and therefore you wouldn't have such severe sickle cell. So this gene therapy trial and subsequent other gene therapy products are using the same hypothesis or the same science that BCL11A, the causative agent of hemoglobin S, if you shut it off or silence it, you would make F, and F would not sickle. Sickle cell gene therapy inserts a gene that knocks down or shuts off the BCL11A induction, thus having persistent hemoglobin F. Sorry. So using this kind of diagram, remembering sickle cell disease knocked down BCL11A gene and the lentivirus. So we put those two together in culture, as we do once we collect the CD34 cells. The cell culture is above. After the cells come out of culture, those cells now have a genetically modified product that when infused, will instruct the body to knock down the BCL11A and continue to turn off sickle hemoglobin and to turn back on hemoglobin F. So the stem cells then will give rise, once engrafted, to RBCs that produce F and not S. And the F circulate and multiply without sickling, which then causes the elimination of anemia and vaso-occlusive crises. These are pain crises. This is just a picture of what that patient's CBC and smear looks like. You can see in the pre-GT, you can see those small non-smears, or those small little, they almost look like a little line, but a little, I don't know the best thing to call them, but you can see they don't look like the spheres. 6 months post gene therapy, if you look, you don't see any of them. So those were the sickled cells, and now those are gone. So this was a patient who, subsequent after gene therapy, no more vaso-occlusive crisis, no more acute chest syndrome, no neurological events, very mild anemia, less red blood cell breakdown, and no transfusion requirement post-engraftment. This is another actually illustration of the steps, though, which is, which it takes to get ex vivo gene therapy. And as you can see, it's actually quite a complex process for the patients, with 11 steps being responsible. So this is how we do it at Boston Children's, but I think this probably depicts how everybody, the steps everybody has to go through. But if you also look underneath in the parentheses, you can see that not only is it 11 steps, but all told, this can take up to a year from the time that the patient has been identified for treatment and consults, all the way through to having the stem cells collected, sending them to a manufacturing facility to have them made, which can take anywhere from 4 to 6 months, and then getting them back, getting them engrafted, and then the follow-up. I will say of note, the follow-up in the United States for the gene therapy, ex vivo cellular-based gene therapies, dictated by the Food and Drug Administration, is patients need to be monitored for 15 years. So in a very brief way of saying, this is not a very simple process, and it takes a lot of commitment, both from the team, from the care team, as well as the patient and family. The nursing care implications around gene therapy are, as just described in the previous slide, it requires a lot of care coordination for a very complex multi-step process and a very multidisciplinary process. It requires the nursing staff to be very vigilant about monitoring and observing for any adverse events. Documentation of the conditioning, infusion of the gene therapy product, and supportive care is all necessary. And then in the case where these are still in the research world or still being conducted under protocol, we certainly rely on our nursing staff on the floor and our nursing colleagues to collaborate with the research team to assure protocol compliance. All the protocol elements have been met, and as I said, all of the documentation about any adverse events or any other elements of the protocol are captured in the medical record. So monitoring for the outcomes of gene therapy, as I said, the FDA mandates that we have a period of 15 years for ex vivo gene therapy to assess for safety, long-term effects, and any late effects. The laboratory testing we do is to check for the vector presence in the cells. So once the cells have been genetically modified, you should be able to see either presence of that vector that transported the gene into the cell nucleus, and or you should be able to see evidence of gene editing or modification through indels. We watch for patients' immune cell recovery because we did use myeloablative conditioning, so we want to make sure that they're immune, that they reconstitute. We can watch for protein expression, and then the cellular immune responses. Patients will have frequent visits and monitoring reports for any adverse events, laboratory abnormalities, and or any unanticipated effects. Just recognizing that although these products are FDA approved, EMA approved, they all still are actually pretty young and pretty new, and have been approved on an accelerated regulatory pathway. So there isn't a lot of history. So maybe less than 10 years. So there could be untoward effects, unknown effects that we don't know about yet. And so that's why we monitor them so closely. And then clinical observations and parental reports for any changes in symptoms, or watching for frequencies of vaso-occlusive events, admissions to the hospital, need for transfusions in the sickle cell population would be important data to understand the efficacy and the outcomes from getting the therapy. And now I explained all the things that are really curative and very exciting about it, but there are things that we worry that could go wrong. One is insertional mutagenesis. So then when we insert the new gene with the vector into the nucleus, you can't put it exactly in the genome where you want it. So it could actually insert somewhere in that patient's genome that could be adjacent to oncogenes or things that could be deleterious to the patient's health. So it could turn on, leading to malignancies. The preventative actions that the field has done around this is new vector designs have been redesigned to reduce the likelihood of activating oncogenes by reducing the intensity of which the vector infects the cells. So as I said, we have harnessed the vector or the viruses so that they permeate that cell and get into the cell membrane and into the nucleus. One of the ways that that was done in the past was they used an enhancer or promoter to drive it faster and harder and a little more regressive. Some of that has been pulled back a little bit so that the vector still infects the cell, but not with its same intensity, but it still has the same efficacy. Another potential problem is, as I said, the stem cells could not take up the vector or fail to edit, leading to no transduction. We do test any product prior to infusion for the presence of the vector. And another safety check is that we never bring a patient in to start conditioning until we have confirmed from the manufacturer and or the laboratory that that cell product that has been genetically modified meets all release criteria, all release specification, including presence of vector. Because we don't want to be in a position where we've conditioned somebody with busulfan or fludarabine, rendering them immunocompromised, and we don't have something to give them back. In the event that happens, for other reasons, that the product could go to the patient but did not engraft for some patient for any reason, and the patient was conditioned, we do have that cell product backup graft that's unmanipulated, which is stored in the freezer, and that we would give back to rescue the patient, but remembering that there was no genetic manipulation. So the intent of having a curative therapy was not met. And then in gene editing, much like the insertional oncogenesis, where the gene would insert or the vector in the gene inserts in the genome in a place that might be dangerous, gene editing also can have that effect. It could edit cells adjacent to where you are editing. Now that becomes a little less problematic as you get into more specific, as I said, with an increasing degree of specificity with base editing and prime editing. That seems to ameliorate that, but that is still a potentially problematic area. And those edited cells are tested pre-infusion to make sure that the cells have been edited, and then they're monitored for the site in looking at their DNA to make sure that there's no there's no misedited around in the adjacent site. I'll stop there. And it looks like we have a question. Is it possible you treat multiple myeloma patients by using CAR T-cell immunotherapy after or before relapse? I don't know I know the answer to that. I can speak to this in B-cell, relapsed B-cell leukemia, which is where we do CAR T-cell in pediatrics. And in full disclosure, I'm actually a pediatric person, so we don't see much myeloma in children. But I will say that I do know that they do myeloma. Certainly have CAR T-cell for myeloma, but I believe it's relapsed myeloma and not frontline therapy. There could very well be clinical trials for frontline therapy. In our relapsed BCLF, for relapsed leukemia, we do Kymriah for refractory or multiple relapse B-cell ALL. All right, well, thank you, everyone. Thank you for your time. Thank you for your attention, and have a nice day and evening. Thank you.